Welcome to our interview questions and answers on Karl Fischer titration. This guide will help you master essential concepts and interview questions for analytical chemistry roles. Question 1. Can you explain what Karl Fischer titration is and what it is used for? Answer. Karl Fischer titration is an analytical method for measuring water content in samples, from trace to high levels. It involves a reaction between water and iodine with sulfur dioxide, a base, and a solvent. This technique is crucial in industries like pharmaceuticals, food, chemicals, and petrochemicals. Question 2. What is chemical reaction of KF titration? Answer. Karl Fischer titration is based on the Bunsen reaction. Iodine is oxidized by sulfur dioxide and water, forming sulfuric acid, which is neutralized by a base like pyridine or imidazole. Methanol acts as the solvent. The stoichiometry is precise. 1 mole of water reacts with 1 mole of iodine. Question number 3. What is difference between volumetric and caloric titration? Answer. Volumetric Karl Fischer titration uses iodine from a burette, ideal for water content above 1%. Coulometric titration generates iodine electrochemically, perfect for trace moisture levels. This method is more precise for low water content, eliminating the need for a standard solution. Direct titration involves adding the sample directly to the titration vessel for immediate reaction with the Karl Fischer reagent. Indirect titration is used for samples that react slowly or interfere with the reagent, involving excess iodine addition followed by back titration. This method is ideal for solid samples with complex matrices. Karl Fischer titration is vital in pharmaceuticals for measuring water content in active pharmaceutical ingredients and excipients. In food and beverage, it controls moisture in powders and processed foods. In petrochemicals, it detects trace water in fuels to prevent corrosion. In chemicals, it ensures quality control of raw materials and products. In Karl Fischer titration, the endpoint is detected electrochemically using a double platinum electrode. Before reaching the endpoint, iodine is consumed by water, resulting in low current flow. At the endpoint, excess iodine causes a voltage drop, signaling the titrator to stop. This ensures high accuracy without relying on color indicators. Interferences in Karl Fischer titration include oxidizing agents like peroxides, which can cause false high water results. Reducing agents, such as ascorbic acid, also interfere by consuming iodine. Carbonyl compounds, like aldehydes, react with methanol, forming water. Address these by using alternative reagents, sample preparation, or techniques like the oven Karl Fischer method. Standardizing the Karl Fischer reagent is crucial for accurate water quantification. The titer indicates how much water 1 milliliter of reagent can consume. Common standards include sodium tartrate dihydrate, pure water injections, and certified water standards. Verify the titer daily or before each batch to ensure precision. For Karl Fischer titration, inject liquids directly into the vessel using a syringe to avoid atmospheric moisture. Dissolve solids in dry methanol or use the oven Karl Fischer method for moisture extraction. For gases, pass them through the titration vessel at a controlled flow rate to ensure moisture absorption by the reagent. The oven Karl Fischer technique involves heating the sample in an external oven, typically between 50 to 200 degrees Celsius. A dry carrier gas transports the released water vapor into the Karl Fischer titration vessel. This method is ideal for samples that dissolve poorly in methanol or contain interfering substances like aldehydes and ketones. Temperature selection is crucial to avoid sample decomposition. Regulatory guidelines for KF titration in the pharmaceutical industry include United States Pharmacopoeia less than 921, which defines procedures and criteria, and European Pharmacopoeia 2.5.12, used for raw material testing. International Council for Harmonization Q6A specifies water content as a quality attribute, while International Council for Harmonization Q2R1 outlines validation parameters like precision and accuracy for KF methods. Method validation for Karl Fischer titration ensures the procedure is suitable and meets regulatory standards. Key acceptance criterion is a water standard recovery of 98 to 102%. Both repeatability and intermediate precision must be demonstrated to validate the method effectively. Drifting baseline in Karl Fischer titration is often due to atmospheric moisture, ensures seals are intact and glassware is dry. Irreproducible results may stem from expired reagents or poor electrode conditioning. If no endpoint is reached, check for depleted reagents or blocked electrodes. High readings could indicate carbonyl interference. Consider using carbonyl-free reagents. In Karl Fischer titration, reagent choice is crucial. One-component reagents, like hydrogenol composite, simplify routine analysis. Two-component reagents offer flexibility for challenging matrices. Modern pyridine-free formulations using safer bases like imidazole enhance safety and regulatory compliance. Choose based on sample type and expected water content. Karl Fischer titration specifically measures water content, making it ideal for volatile samples and regulatory submissions. It's accurate to the parts per million level. In contrast, loss on drying measures total mass loss, including solvents, and is simpler and cheaper but less specific. Choose Karl Fischer for precise water data. Use loss on drying for quick, non-critical checks. For interviews, grasp the stoichiometry. 1 mole of water equals 1 mole of iodine. Differentiate between volumetric and coulometric methods and direct versus indirect approaches. Highlight your practical knowledge in standardization, troubleshooting, sample preparation, and regulatory compliance to distinguish yourself.